Okay, so the next moderator will be Dr. Nikmatuzaro from Erlangga University.
Okay, thank you, Ibu Daya, for the time given me. Good afternoon, ladies and gentlemen. Now we enter the third parallelization in the class A with the topic of biotechnology. I want to introduce my name. My name is Nikmatuzaro. I came from Universitas Airlangga, and this afternoon I was asked by the committee to be the moderator for this parallelization in the class A. In this session, there will be 4 topics to be presented. Okay, we will check first. Are all the presenter present now? Okay, I saw the names of the representative of the 5 articles were already present. And remember that we have 1 hour in the session 3 with a 4 presenter, so that each presentation time is 10 minutes. And the remaining 10 minutes we will use for discussion. Okay, now I want to invite the first presenter. The topic is the effect of phosphate concentration and ammonium and nitrate ratio on the biomass of Java ginseng, adventitious roots in the culture. Please take your time, Ms. Putri Kiasti. Yes, okay.
Oke, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you for the opportunity. My name is Putri Gahasti. I'm from Biology, Faculty of Science and Technology, Erlangga University, Surabaya. Today, I'm going to present my research. The title is The Effect of Phosphate Concentration and Ammonium Nitrate Ratio on Biomass of Java Ginseng Adventitious Roots in Vitro Culture. The topics consist of introduction, material and method, result and discussion, and conclusion. The first one is introduction. As we know that plants are an incredible source of novel medicinal products invention to develop, and one of the medicinal plants that are often used by the community of Java ginseng or Thalinum paniculatum. Roots of Java ginseng majority used as medicinal ingredients because Java ginseng has potential as antimicrobial, anti-inflammatory, and immunomodulator agents such as saponins. Thalinum paniculatum has a slow root growth and its natural habitat, since it takes 2 until 3 years for producing 100 gram roots per plant. So we need to the new technique for increasing the biomass of roots of Thalinum paniculatum. In vitro culture techniques poses to be an alternative way to accelerate the growth of the roots of this plant. Root growth can be increased by manipulation in culture media with the addition of nutrients, addition of precursors, and optimization of environmental factors. So in this research, we adding some concentration of phosphate and nitrogen. That phosphate and nitrogen play an important roles for plant growth, especially for energy transferring, DNA and RNA synthesis, and protein formation. So the aims of this study is to see how the effect of phosphate concentration and ammonium nitrate ratio in culture media on the root biomass of Thalinum paniculatum adventitious roots. Next is material and method. The first one is adventitious root induction from leaf and stem explant. The adventitious roots of Java ginseng were induced from stem and leaf explant on solid MS medium with various combination of growth hormone, EBA, 2 mg per liter, and combining with various growth hormone of cytokinin. There are BAP, kinetin, and tidiazuron. And then leaf samples were used, the leaves taken and third from shoot, and all explants were sterilized using detergent solution and 10% of Clorox. And then all the explants were maintained in temperature 25 Celsius in dark condition, and after 28 days, adventitious roots were harvested, and measurements of fresh weight, dry weight, number of roots, and duration of adventitious roots were measured. And second is preparing of addition phosphate and adventitious root growth. The explant used the leaf second and third from shoots and then solid MS medium supplement with IBA 2 mg per liter and adding some different concentration of phosphate. After that, leaf explant were incubated at 25 Celsius in the dark for 6 weeks. And after 6 weeks of cultivation, the number of roots, fresh weight, dry weight, root length, and root formation time were measured. And then preparing of addition ammonium nitrate ratio on adventitious root growth. The first is the explant used the leaves, second and third from shoots, and then MS medium was supplemented with IBA 2 mg per liter with different ammonium nitrate ratio. And then leaf explant were grown in medium and incubated at 25 Celsius in the dark for 6 weeks. And after 6 weeks of cultivation, the number of fruits, fresh weight, dry weight were measured. And then for the data analysis, the data obtained include fresh weight, dry weight, number of adventitious root, and length of adventitious root were analyzed using ANOVA at a significant level of 5%. The next is result and discussion. This is the figure of Thalinum paniculatum adventitious root induction. Figure 1 is using stem explant, and figure 2 is using leaf explant. This is the table of the result that the best treatment of producing biomass of roots Thalinum paniculatum is in treatment with supplement IBA 2 mg per liter combined with kinetin 0.3 mg per liter. And the table 1 is from stem explant, and table 2 is from leaf explant. That the best treatment is producing a higher biomass is in treatment IBA 2 mg per liter combining with tidiazuron 0.1 mg per liter. After we see the result, we know that the selection of types, concentration, and combination of growth regulator is very important. Kinetin hormone can affect the process of plant development at low concentration. Meanwhile, tidiazuron can stimulating endogenous cytokinin production, and IBA can play roles in the process of cell division and enlargement, especially of fruit formation. And the effect of this growth regulator is depend on the type of plants and the concentration of plant growth regulator combination, which is suitable for plant growth. And this is the result of the effect of phosphate concentration, that the best treatment that producing the highest biomass is in P5 treatment, that the concentration of phosphate is 340, that the highest fresh weight, dry weight, and number of roots is in P5 treatment. And this is the figure of adventitious root after cultivation with phosphate. This one is the best treatment. And then this is the table of the result of the effect of ammonium nitrate ratio on adventitious root growth. That the best treatment is ratio of ammonium 10 and nitrate in 20. And this in this ratio, they can produce the highest biomass of fruit Thalinum paniculatum. This one figure that C is the best treatment. That we know that nitrogen is used for protein synthesis, so it is needed for cell and organ growth, including the production of plant biomass. The form and amount of nitrogen in the in vitro media have a significant effect on cell growth rate, differentiation, and cell totipotency. So this one is the conclusion. The first one is media MS supplement with IBA 2 mg per liter combined with kinetin 0.3 mg per liter is the optimal concentration to produce the highest mean number of fruits, fresh weight, and dry weight of Phthalinum paniculatum adventitious root in stem explants. And then the best result for the leaf explants were obtained by adding IBA 2 mg per liter combining with Lidiazuron 0.1 mg per liter. And then the highest fresh weight concentration were obtained in the concentration of phosphate is 340 mg per liter treatment. And the last one is the ratio of ammonium nitrate 10, the ratio of ammonium 10, and the ratio of nitrate 20 was the best treatment to produce the highest biomass. Thank you.
Thank you very much, Ms. Putri Kehasti. I invite the second presenter. Please, for the Miss Nur Hidayah, take your time.
Okay, thank you. But I can the
Yes. I make you a host now. Okay. Please share your PPT. Yes.
Okay, thank you. Okay. Okay, thank you, Miss, for the time. The Excellency, the keynote speaker, the chairman, chairperson, the committee, and all of the presenters in this conference. In this question, I will present about the Simplicia production, extraction, and identification of bioactive compound from the leaf of peach morning glory or Ipomea pescaprae from the coast of Asam Doyong, Pemalang, Nur Hidayah, and then the Dede Purwanto and Siti Sarwanurbani. And then this is the content of this presenter. One is introduction, material and methods, result and discussion, and the last is the conclusion. I will start with the introduction. We know that Indonesia is an archipelago country with a long coastline, and Pemalang is a northern area of the Java Sea. We know that Asam Doyong is one area in the Pemalang, and its beach that has characterized the sandy coastal area. The sandy coastal area are more dominated by the plant with the pescaprae formation, like Ipomea pescaprae or beach morning glory. And the beach morning glory or Ipomea pescaprae have characterized that this plant has a flower like a trumpet with the purple flower. And this plant have potentially aggressive invasion and blooming. It's because there are no pests or diseases that are serious enough to infect these plants. Growing of the Ipomea pescaprae need full of the sunlight, and the leaf of this plant are thick, smooth surface, and have two lobes. And about the roots occasionally branches from the nodes and develop long, thick, and stretchy root. Several studies have said that the beach morning glory or Ipomea pescaprae contain a chemical compound like alkaloid, saponin, tannin, and alpha-flavonoid. The bioactive compound are secondary metabolites that to defend themselves from unfavorable environment conditions. The bioactive compound like alkaloid and saponin that have a pharmacological effect. The alkaloid exhibit hypoglycemic activities, and the saponin as antioxidant and also the anti-inflammatory. Therefore, it's necessary to us to produce the Simplicia from the leaf of the Ipomea pescaprae, and then the extraction and identification the bioactive compound from the leaf of the beach morning glory or Ipomea pascaprae, the ground on the coast of Asam Doyong, Pemalang. This is material and methods. The material that we use in this research, such Ipomea pascaprae leaves, some solvent, and several reagents such as Mayer, Dragendorf, and Wagner. And we have to use this is a breeder, and then orbital shaker, rotary vacuum evaporator, and etc. This is some methods. The first, we we have a sample preparation. In this stage, we pick the leaf of the Ipomea pascaprae, and then we morphometric analyze, and then we get the second stage. It's the Simplicia production. We dry the leaf, and then we get the powder of the Simplicia. And then the third stage is the extraction. We extraction with the 3 different solvent with different polarity, like N-hexane, ethyl acetate, and the ethanol. And then we have some analysis method. For Simplicia, we have water content and as analysis. And for the crude extract, we doing the alkaloid and saponin test. This is the scheme for Simplicia production. We start with a sample preparation, and then drying the sample, drying the leaf of the Ipomea pascaprae at the room temperature, and not exposure to the direct sunlight. And then after that, we get the Simplicia powder and analyze it with the water content and ash content. And this is the scheme of the stratified extraction, where is the Simplicia powder macerated with the N-hexane solvent for 24 hours, and then we filtration to get the residue and the filtrate. The filtrate, and then we evaporation with the rotary vacuum evaporator to get the N-hexane crude extract. And the residue, we macerated again with the ethyl acetate solvent for 24 hours. And then we filtration to get the residue and the filtrate. The filtrate we evaporated again with the rotary vacuum evaporator to get the ethyl acetate crude extract. The residue, when then we re-macerated with the ethanol solvent for 24 hours, and then we filtration get to get the residue and the filtrate. The filtrate we evaporated with rotary vacuum evaporator to get ethanol crude extract. And then for phytochemical test, for 3 extra we have N-hexane crude extract, ethyl acetate crude extract, and ethanol crude extract. We get analysis to alkaloid and saponin test. And this is the result and discussion. This is the area of the Asam Doyong beach that is this location geographically located at this coordinate, and the population of Ipomea pascaprae or beach morning glory. This plant require direct full sunlight to carry out the photosynthesis. The Ipomea is an evergreen plant that live on sandy coastal area and very tolerant to high salinity. This plant is have rapid growth. Its rapid growth has made the population of Ipomea pascaprae in Asam Doyong is quite tight. The Ipomea pascaprae has morphology and morphometric. He has around cylinder shape. He has a cylinder shape and with a green color, and has a segment. He has a segment for node along the stem. The leaf have a facial attack to each base of the stem segment. And then the leaf are dark green, thick, smooth, and have 2 lobes. From the morphometric measurement, Epimeth pascaprae has a length for 4.5 centimeters and width the 8 centimeters. The leaves used as the Simplicia are healthy and clean leaves. They are not contaminated with the disease. We just pick the leaf to maintain the sustainability of the Epimeth pascaprae on the coast of Asam Doyong. And then this is a Simplicia powder. The Simplicia powder we get from the 4 kilograms of fresh Epimeth pascaprae leaf, and we get 360 grams of Simplicia powder. So the yield of this Simplicia powder is 9.05%. And we have analysis of this water content and the ash content. The water content is 5.65% and the ash content is 18.51%. And this is the result of the crude extract. The crude extract have a texture like a paste, and it have a dark green color. And then we have identified the bioactive compound and then from the 3 solvent we have extracted, we have a higher yield in ethanol solvent for 5.62% of crude extract. And this is the phytochemical test for testing the alkaloid and the saponin. From this result, in alkaloid test, so the negative result for 3 extract and for 3 reagent, except the Mayer for ethyl acetate extract. And for the saponin extract, for the saponin test for 3 extract have showed the positive result for the extract and hexane and the ethanol extract, and give the negative result for the ethyl acetate extract. And this is the last of the presentation about the conclusion. The Simplicia produced from the leaf with size of 8 in width and 8.5 in length. And then dry powder Simplicia have yield of 9.05% with water content 5.556%, and the ash content is 18.51%. The high yield of the crude extract in ethanol extract that is 5.62%. And the 3 extract show the negative result for the alkaloid test, and for the saponin test show the positive result for N-hexane and ethanol extract. Get the negative result for the ethyl acetate extract. Okay, thank you.
We call again to Miss Ellie Trivijayanti. Now you are present. Please take your time for your presentation. Hello.
I'm sorry, Ibu. Mrs. Ellie is just come in. Maybe she hasn't got to be co-host.
It's okay, Miss Ellie. Yeah. Now Miss Ellie is a co-host.
Hello, Mom Nikmah. I am
Yes.
Ellie. I'm here.
Yeah.
Yes.
Your 10 minutes only, yes?
Yes. Okay. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Yeah, this is Ellie. I'm from Erlangga University, Biology Erlangga University. This is I want to presentation about our research by title Enhanced Biomassa Production and Saponin of Thalinum paniculatum adventitious root in balloon type bubble bioreactor use batch and feed batch system. Okay. This is introduction. So the Javanese ginseng or Thalinum paniculatum was known as Thalisum, Colesum, or Javanisum is classified into the Portulaceae, one of secondary metabolite that accumulate in the roots of Thalinum paniculatum was saponin. The root of Thalinum paniculatum were known to be effective in overcoming sexual dysfunction, irregular menstruation. Yes.
Hello. Please slideshow, please.
Oh, sorry.
Yeah.
Yes. Okay.
Yeah.
Okay, root culture in a bioreactor is an expected method for producing large amount of root biomass and secondary metabolite. The most frequently used bioreactor was the balloon type bubble bioreactor or BTBB. This research was carried out to improve the culture condition of Thalinum panicunatum adventive root in small-scale BTBB by mixing some modified that is adding of aqueous without a new medium. We call batch and adding new medium periodically or feed batch to reduce some constraints that appeared in previous study. Induction of adventive root, the leaf were taken from the Thalinum paniculatum plant. It was with a detergent solution for 10 minutes, rinse under running water, and then the leaf were cleaned again using 10% Clorox solution for 10 minutes, then rinse with sterile distilled water 3 times. It was cut into 2 centimeters size. The leaf piece were transferred to Petri dish, which contain solid medium MS and 2 ppm IBA. And then culture were maintained in incubation root at 25 degrees Celsius without tightening. And the last, after 5 weeks, the adventive root which were taken 2 gram per 3060 milliliter as the initial inoculum, transfer in Erlenmeyer containing the same medium as before to be shaken for 36 hours at 100 RPM. This is design of BTBB. This is A is a balloon reaction vessel. In B, inlet for the media and inoculum. This is C. This is a media transfer channel. D, pH buffer channel. E is aeration pump. F is rotameter. G, air intake. And then filter, air outlet, bubble breaker. This is bubble breaker or sparger. And then K is media outlet for sample. And this is wear netting. The accumulation adventive root were transferred to liquid MS media in BTBB 1 liter. Each bioreactor was given an inoculum density of 2 gram adventive root and an aeration rate of 0.2 ppm. Bed system, there was no add new medium but distilled water periodically and was given or not a pH buffer to make a combination of batch buffer and batch not buffer as control. And feed batch system. Feed batch system is the bioreactor was add periodically with a new medium without distilled water. Then the bioreactor was given or not a pH buffer to result a combination of feed batch buffer and feed batch and buffer as control. The research variable which observed and measured were fresh biomass, dry biomass, growth speed, saponin concentration. Saponin concentration is measured by TLC, and then physical media. There are medium pH, total sugar content, electrical conductivity. Determination of a adventive root at saponin content. Saponin content was determined using TLC and TLC densitometry method. 100 of dry biomass powder dissolved in 10 milliliter ethanol and then heat at 18 degrees Celsius in water bath for 3 hours or until it become concentrate liquid to 0.2 milliliter. And then the extract was put on silica gel and elute using propanol and water ratio. Its ratio 40 and 3. And then the presence of saponin compound was compared to standard saponin. And then the silica gel plate was sprayed with 0.5 milliliter anisaldehyde sulfuric acid stain. And then it was tested and analyzed against semi-qualitative using TLC densitometry. This is adventive root biomass in 4 treatment. There are feed batch buffer, feed batch unbuffer, batch buffer, and batch unbuffer for 5 weeks in a bioreactor containing liquid MS at IBA to ppm. This is morphology of adventive root, Talinum paniculatum. E until D, this is biomass in batch, batch plus buffer, feed batch plus buffer. E until H, this is dry biomass in batch, batch buffer, feed batch, and feed batch plus buffer system. Okay. This is visual comparison of stain of the LC plate from in vitro extraction of Talinum paniculatum root. Number 1 till 5 is a standard solution of saponin with concentration of 0, 10, kemudian, and then this is for treatment our research. This is retention factor, compound concentration and saponin area from the extract of the adventive root of Talinum paniculatum. The table show that buffer, batch and feed batch not detected of concentration of saponin, but unbuffer batch or feed batch show the concentration of the saponin. This is change in pH, electrical conductivity, and total sugar content during Talinum paniculatum adventive root culture on bioreactor. Minus for before at buffer and plus for after at buffer. So the conclusion of our research is the production of biomass of Talinum paniculatum adventive root were obviously higher in the feed batch buffer system than other treatment. The system 4 times more from initial fresh white after 35 days culture. Supplementation of buffer, we use sodium bicarbonate, maintain the condition of the media in the bioreactor. So as they provide the highest biomass in a feed batch system, but the maximum amount of saponin occurred from the batch system that 2.30 milligram per 100 milligram of saponin concentration on dry weight. Okay.
Okay, thank you. Thank you, Miss Ali. This is a nice presentation, I think. Okay, we go now in the fourth presentation from Miss Dita Arya Widyatama.
Yes, madam.
Yes, okay. Take your time, please.
Okay. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Yes, thank you for the opportunity. My name is Dita Arya Witatama. I'm from Gajah Mada University. Here I will present about my research. The title is Zebrafish embryo immune system induction with BSA as initial screening model for COVID-19 treatment. Dita Arya Witatama and Bambang Ratno Aji as my corresponding author. So, the vaccine are antigen that apply a raw body immune system. Immune system consists of innate and adaptive immune. Adaptive immune can be induced and form memory cell. Vaccine induction, vaccine induction several types of antigen which protein. BSA or bovine serum albumin is a protein that can function as a tracer related to antibody and memory cells. Administration of BSA at the beginning of embryo development can interfaces with formation of 3 germ layers, can causing abnormalities in embryo development. Administration of certain dose at the beginning zebrafish embryo development was expected to affect the formation of immune system. The treatment with junior extract and its effect on age population is a COVID-19 receptor in future study. Zebrafish is appropriate for use in this research because it gets their oxygen supply through passive and transparent and also have various genes in humans, such as an S2 receptor. This study is a screening study in order to prepare the right BSA dose to influence the zebrafish immune system in the context of COVID treatment. The study in initial screening to prepare the right PSA dose to influence the zebrafish immune system in the context of COVID-19 treatment. My material and method. Material is zebrafish BSA or bovine serum albumin, egg water, and equipment is silica microscope and petri dish and pipette. There are 3 step in my research. The first is first setting, 3 female and 6 male wild type zebrafish were maintained on well-aerated aquarium for 2 weeks prior mating. And the egg collecting fertilized fish eggs were collected and inspected under microscope to select and ensure only normal and good quality of zebrafish egg. And the third egg were immersed in 1%, 2.5%, and 5 for the period 5 hours with 3 replication each. The analysis is muscle contraction, heart rate, and survivability. The data obtained were the staged by ANOVA post-hoc TMRT with a 95% confidence level. Data analysis was performed using SPSS software. This is BSI effect on early stage embryo. Early stage of embryo exposed to different concentration of BSA on 24 hours. The BSA effect on embryo survivability. Embryo survivability were decreased from the concentration of 1% to 5%. The lower survivability of embryo on BSA 5%, 5 hours and 48 HPF. The results suggest that 5-hour BSA exposure caused permanent change on embryonic development, since it was exposed on the period of cellular differentiation and organ formation of the embryo caused defect on organ structure and function and leading to fatality. Our exposure of 5-hour immersion, early development embryo really on communication between cell and affected. Chemical signal BSL was made from bovine co-embryo that rich in growth factor that affect cell communication easily and cause certain defect. BSL has the ability to ligand binding also as various transporter. BSL ability on ligand impact on non-specific binding protein in genes. BSA effect on protein channel opening is called enhanced calcium concentration in cell enhance of calcium concentration cause muscle concentration enhance. The figures to show zebrafish embryo exposed to different concentration of BSA on 24 HPF for our post-fertilization. This is BSA effect on heart rate. Results show that the highest heart rate was on 18 HPS BSA 5%, 5 heart immersion. The lowest on 48 HPS BSA 5%, 5-hour immersion, also 2.5% and 5%, 5-hour immersion were died. Concentrations of BSA cause defect in osmotic pressure, which result cause defect embryo develop and several states. The effect of BSA concentration of zebrafish embryo we also did in the chorionet ad for comparison. The figure 4 shows zebrafish. The figure 4 shows zebrafish embryo exposed in different concentration of 48 HPF of BSA. This is the decoriented zebrafish embryo that has been immersed in BSA. BSA show a lethal effect which resulted embryonic death after somit formation phase, while the non-decoriented zebrafish embryo can survive until hatching. This indicates that the chorion has an important protective function to filter out non-permeable substance from the embryo. The figure 5 shows the effect of BSA concentration on zebrafish embryo on the decorionate egg. The conclusion is BSA, or bovine serum albumin, has significant effect on normal development and organ function of zebrafish embryo, such as muscle contraction and heart rate of the embryo. This suggests that BSA is a good candidate for the zebrafish immune system induction for the further experiment. This is my reference. Thank you.
Okay, thank you, Ms. Gita. Please
Yes.
stop your PowerPoint, please. Now,
Yeah.
we are in the last presenter. We invite to Mr. Arif Lukman to present your article entitled "The Effect of Tryptamine on the Gram-Negative Bacteria." Please take your time, please.